Our next speaker is Joyce Lee from San Francisco, who's going to tell us about telomere length, disease severity, and survival in IPF.
Yes. Great, thank you. So as many of you in this room know, short telomeres are very common in IPF. They have been observed in both sporadic and familial forms of this disease, and they've been measured to be short in both the alveolar type 2 cells as well as the peripheral blood leukocytes of patients with IPF. And what you can see here, in this figure, this is the telomere signal per cell, and in normal patients compared to sporadic and mutation carriers, and you can see that the telomere signal per cell in the alveolar type 2 cell is lower in both sporadic and IPF mutation carriers. In addition, the cause of the short telomeres in patients with IPF is not entirely clear, but genetic mutations have been described in a subset of patients. So mutations involving telomerase and its RNA template TERK have definitely been described in some familial forms of IPF. And the thought here is that these short telomeres may contribute to the pathogenesis of IPF by promoting alveolar epithelial cell senescence and apoptosis. So basically, that proposed pathobiology is described here in this diagram. And what is thought is that short telomere length here, whether it's you get there by a genetic predisposition, a mutation, or some environmental cause, will lead to increased alveolar type 2 cell senescence and cell death, which leads to pulmonary fibrosis, which ultimately leads to death. And very simply, my question was, is short telomere length associated with death in IPF? And my hypothesis was that shorter telomere length is associated with worse survival in IPF. This is the study design. So we have patients who've been prospectively enrolled into our database, and they have, a handful of them have DNA collected at the time of diagnosis. The primary outcome in the study was death or lung transplant, and the covariates that I had available and measured included age, gender, body mass index, smoking status, symptoms of cough and dyspnea, pulmonary physiology with FVC and DLCO % predicted, radiologic fibrosis score, medications, and oxygen use. The telomere measurement was provided by a company called Telome Health. This is a company started by Elizabeth Blackburn from UCSF, and she provides this service to researchers as well as actually the community to have your telomere length measured. And the telomere length is that they provide for you is a quantitative PCR method. It's economical, versatile, and it's a validated high throughput method. And what it generates is a TS ratio, which is essentially proportional to a cell's average telomere length. And just to describe this a little bit further, I know there's a lot of words here, but very simply, the TS ratio is essentially the telomere length. And it determines the ratio of telomere repeats to a single copy gene in an experimental sample. So there's a reference sample that Telome Health uses, and so it's, you can't really translate the findings to another lab who has a different reference sample. So the T signal is the number of nanograms of the reference DNA that matches the experimental sample for the copy number of telomere repeats, and the S signal is the single copy gene. And so if you have a TS ratio greater than 1, then that person's telomere is longer than average compared to the referent DNA, and if it's less than 1, it's shorter than the referent DNA. And it correlates very well with the traditional Southern blot technique. So here's the data. There were 59 subjects with IPF and had DNA collected at the time of diagnosis. The average age was 69. The majority were men with an average body mass index of 28. As expected, most were former smokers, and 90% reported cough. The mean dyspnea score based on the CRP scale was 9, which essentially equates to feeling short of breath after approximately a half a block. Pulmonary function was as expected. 71% was the mean FVC % predicted, and DLCO was 46% predicted. A quarter were on prednisone at the time of diagnosis. 5% were receiving azathioprine, and 7% were receiving NAC. 17% were already on oxygen use by the time they saw us. The median radiologic fibrosis score, which was only available in a subset of these patients, was 14%, and 49% had lung biopsy as part of their diagnosis. So this is the telomere ratio in our cohort, and the purpose of this slide is just to show you that the telomere length is essentially normally distributed within this population, with a mean of 1.04 and a median of 1.06. So looking at measures of disease severity, we found no association between telomere length and FVC percent predicted and DLCO percent predicted at baseline. What you can see here on the x-axis is your TS ratio, and then on the y-axis for this plot is FVC percent predicted, and on the right one is DLCO percent predicted. We did, however, find an association between radiologic fibrosis score and telomere length. So the higher your radiologic fibrosis score, the shorter your telomere length was. So now I'm moving along to our survival analysis. And so I've looked at the survival data in several different ways just so that we can better understand what it might mean. So arbitrarily choosing a cutoff at the mean TS ratio of 1.04, I dichotomized the variable. You can see that patients with a longer telomere length in this dashed line here live longer than patients with a shorter telomere length. Looking at the data a little bit more effectively using telomere length as a continuous variable, what you can see here on unadjusted or bivariate analysis, the hazard ratio is 0.19 with a p-value of 0.09. And what this means essentially is for every standard deviation unit decrease in your telomere ratio, the hazards for death increases by 42%. And if you adjust for the things that you might expect would either impact telomere length and also the outcome, we adjusted for age, gender, FVC, and DLCO percent predicted. The hazard ratio again remained very low with a p-value of 0.045. Because the data are a little limited in terms of the sample size being small at 59, I did look at the survival analysis in multiple different ways. And what you'll see in the gray boxes here are the original survival analysis. So transplant-free survival was the primary outcome, unadjusted analysis here with the hazard ratio and the p-value, and multivariate adjusting for age, gender, and pulmonary physiology here in the second gray box. I looked at adjustment for the gap model, which, as you all know, recently published this year, looking at as a predictive model for IPF. And so if you put the gap model in with the telomere ratio, it still remains strongly predictive of survival. If you look at time to death and sensor at lung transplant, again, you can see all of the hazard ratios are in the same direction with varying p-values due to a number of events. And then finally, using a slightly more sophisticated statistical technique, there's a method called competing risks. And so you don't treat death and transplant equally, but you look at transplant as a competing risk for death because you can't die and get a lung transplant. So it's almost like an adjustment factor. And so if you look at it, you can get a transplant and then die, but you can't get to the death first. But if you look at it this way, again, all of the hazards ratios are quite strong in a protective manner, and the p-values, although not all significant, are in the trend towards significance. So in summary, there's a relatively normal distribution of peripheral blood telomere lengths in IPF. Shorter telomere length in this cohort appears to be associated with increased radiologic fibrosis score and shorter survival time. So future directions for this work, we're working on validating these findings in an independent, larger cohort. And there's potential to explore the role of telomere length as a biomarker, both for screening and diagnostic purposes, and also to explore the role of telomere length in other interstitial lung diseases. And this is the group that helped me with the project.
Dr. Lee, did you check for TERT and TERT mutations in this group of patients?
I have not yet.
Have not yet. And my second question is, your control group was age matched and sex matched, gender matched?
There is no control group in this study.
But I want to ask you about a control group of patients at this age in terms of the TS ratio.
Mm-hmm. There should be one done to measure that, and we're in the process of collecting healthy controls, correct?
Okay.
So do you think Gisli Jenkins from Nottingham, do you think...
Would you identify yourself, please?
Yeah, so Gisli Jenkins from Nottingham. Do you think telomeres change over time? So do you think, and that may relate to the progression of the disease, so if you could do longitudinal studies?
I think that's a fantastic idea, and so it would be fascinating to see, multiple time points of telomere length over time and see if it's the change in telomere length more than the baseline telomere length at the time of diagnosis, which can be quite arbitrary, if that's more predictive of outcome than the baseline length.
Okay, we'll have a question from David, and then we'll go to the other room.
Okay.
David Schwartz from Denver. Very nice study. It impresses me that organs age at a different rate, and I was wondering whether anyone has looked at telomere length in different organs and how that compares to each other within an individual. And also, specifically for IPF, whether anyone's looked at telomere length in the lung as opposed to peripheral blood.
Yeah, so I can answer your second question much easier than I can answer your first one. So yeah, the original figure that I showed on my first slide, those telomere lengths were in alveolar type 2 cells of patients with IPF, so it is in lung tissue. In other diseases, I am not familiar with any literature looking at telomere length, for example, in the pancreas or something like that, but I do, I don't know the literature in that regard.
Okay, let's go to the outside room, and then we'll come back in here. Go ahead. Identify yourself.
Yes, I am Moises Selman from Mexico.
Oh, I thought.
No. Moises, I don't want you to talk yet.
Oh.
I want him to talk, but he's not. The sound is off, so we'll go to Moises. Go ahead.
I would like to make a general comment about the shortening of telomeres and IPF, or pulmonary fibrosis. I have some mixed feelings with this idea because mice with telomerase mutation develop emphysematous lesions, not fibrotic lesions. Mm-hmm. They are resistant to bleomycin-induced pulmonary fibrosis, even with very shortened telomeres. And in COPD, you also have shortening of telomeres with, including in the epithelial cells. And on the other hand, telomerase have another function that we know now. For example, it's able to increase the expression of Wnt and exaggerate the Wnt pathway. So I am not sure if we are in the correct way in the pathogenesis of IPF. I think that this study is very interesting because more shortened you have the telomeres, perhaps you have a worse survival. Right. This check with my logic, but not with the general concept of the biopathology of IPF.
I absolutely agree with you, and I think this type of study doesn't get at, I think someone made a point earlier today about chicken or egg. So is the telomere short because it's first short and causes the pathogenesis of IPF, or is it short as a result of the pathobiology that's going on in IPF lesion? Yeah, I agree.
Okay, can you...
From outside now? Yeah. So this is Ian Glasspool from Australia. I just was wondering about whether telomerase or short telomerase predicts for respiratory death, or is it just a risk factor for death in general? What were the causes of death in your study?
Yeah, so that's an excellent question, and unfortunately in our database we don't have cause of death, but generally we think...
Sorry.
That's okay. We think that most of the cause of death is progression from respiratory failure, but honestly I couldn't tell you. It could just be a general marker for for badness, and it just, if you have a short telomere length, you're just not going to do very well. So it doesn't identify what cause of death.
Okay, any other questions? Thank you, Dr. Lee.
Okay, thank you.